So hello to everybody. Thank you also for the introduction. So I'm very glad to be here. And so I'm Laure Sayas. I work for On Beauties. We are a French company specialized in the evaluation of biological efficacy of cosmetics. And for today, I will speak about some molecules called Specialized Pro-resolving Mediators that are part of a superfamily of ligands involved in skin biology. So let's start with what really matters. Why do we use cosmetics? We use cosmetics to improve skin beauty, for example, for skin healing, hair care, sensitive skin, aging, pollution, oily skin, and acne. But what is the common point between all those skin troubles? And be prepared, because there I will leave a bomb. Inflammation. So I already feel some people here in the audience feeling a bit uncomfortable with this word. But I think that this word is really important to be better understood. What is inflammation? I actually read a very interesting article recently. It was published in a web journal called Cosmetic Design. It was published last November, and it was entitled "Why the Approach to Skin Inflammation Needs Careful Thought." And I read this very interesting sentence saying inflammation is a natural response to the immune system to protect the body from harm, and inflammation is a vital part of a body's. It means inflammation is protection. Inflammation is a way to protect the body. It can protect the body from all those skin troubles that I previously mentioned. But of course, what is important is to be able to control inflammation and restore homeostasis in tissue. And this is what is called resolution. So what is resolution? Resolution is a biological process to maintain skin homeostasis. So here in this picture, I represented in a very simplified way the inflammation intensity along time. So inflammation is usually divided in three main steps. We have a first step of initiation, which is due to an irritation, UV exposure, infection. Then there is a step of amplification. This step is characterized by the production of specific biomarkers, mediators, as pro-inflammatory cytokines, chemokines, prostaglandins, thromboxane, leukotrienes, all those mediators that will recruit the immune cells on the inflammatory site. And then there is a return to homeostasis. And this phase is called resolution. Resolution is an active process. It means that it needs the active production of some mediators to induce this return to homeostasis. And this is what is called Specialized Pro-resolving Mediators, SPMs. There are four main families of those mediators that are called lipoxins, protectins, maresins, and resolvins. And so this is the active production of those mediators that induce the return to homeostasis. So those molecules, those SPMs, are lipids, and they have been broadly studied for the last 20 years. And they showed, well, you can find a lot of scientific literature on the subject, and you will see that they have a lot of beneficial capacities as, of course, to decrease inflammation, also clean the inflammatory site as increased elimination of microbes and elimination of dead cells. But they will also participate to the modulation of the adaptive immune response. They will allow a better wound healing by avoiding fibrogenesis and also avoiding chronic inflammation. And of course, reduce pain. Where do they come from? So those molecules are lipids. They are coming from some fatty acids that you know, omega-6 and omega-3. Those fatty acids are present naturally in the cell membrane. So for example, we have arachidonic acid, DHA and EPA, and those lipids are metabolized by various enzymatic pathways to produce some intermediates, and then there is a second step of enzymatic reaction to produce the final mediators. So in the red box, you can find the pro-inflammatory mediators, prostaglandin, thromboxane, leukotrienes. And in the green box, you can find the pro-resolving mediators, so the 4 families I previously mentioned. Lipoxins, maresins, protectins, resolvin D from DHA, and resolvin E from EPA. And of course, I'm speaking about all those mediators because we have the technology to quantify them by analytical chemistry, so LC-MS. Now the question is, how do they work? How do they act? Those SPMs are lipid mediators, and they interact with some specific receptors. And here in this review, they mentioned 6 receptors that can interact with those SPMs. These receptors are GPCR, so G protein-coupled receptors. And of course, once they have activated the receptor, then there is a signal transduction that will lead to the activation of resolution. So, in other words, to summarize a bit what I said previously, when we consider some skin troubles as irritation, oily skin, aging, pollution, itching, all those troubles have an inflammatory component that is due to the imbalance between pro-inflammatory and pro-resolving mediators. And your product, your ingredient, can actually restore this balance by activating resolution and restoring the balance between those 2 types of mediators. Probably your question will be, which ingredient can induce resolution? It can be oils, soluble compounds, herbal extracts, botanical extracts, algae, marine extracts. And of course, we have at Ambiotis all kinds of experimental models to assess the biological capacity of your compound or about your product to activate resolution. So, for example, we can use human primary cell culture models with immune cells and skin cells. We can also perform co-culture. Then we can implement 3D models with reconstructed epidermis and skin explants. And we will stimulate those skin models and then quantify the production of those lipids by our LC-MS device, which is a lipidomic analysis. And we can also quantify those lipids in some samples collected on volunteers. So, for example, on skin strips or swabs. And I will finish with some scientific facts. So here, for example, in this article from Nature Report, they showed the activity of maresin 1, which is one of these SPM. So the activity to protect the skin from inflammation caused by UVB irradiation. And particularly, they showed that maresin 1 can decrease TNF-alpha and decrease MMP9 activity. Also, here are some publications where we participated, where Ambiotis participated. So we showed, for example, the link, well, the impact of resolution in some dermatological troubles as psoriasis and atopic dermatitis. And we also showed the link between resolution and skin aging, as there is a defect of resolution in aged skin. And finally, we also quantified some mediators in samples collected on volunteers. So there, particularly on swab of oily hair volunteers. And we showed that the product can decrease some inflammatory biomarkers as IL-8 or PGE2, but increase some of the lipid mediators from a resolution. So, for example, lipoxins and resolvins. So I hope that this presentation will have given you some new ideas for your future studies, and I thank you for your attention.
Thank you very much for your wonderful talk.